we go. Here we go.
Human cells do not have enough DNA to have separate genes for each antibody molecule. Instead, different segments of DNA can be mixed and matched to form different antibodies. Light chains are made from V and J segments in the variable region and a constant region segment, whereas heavy chains are made from V, D, and J segments in the variable region and one of 5 different constant region segments. As the lymphocyte divides during maturation, its genes are rearranged. DNA sequences are assembled by the random selection of V and J gene segments for the light chains and V, D, and J segments for the heavy chains. The constant region of the molecule is encoded by a single C or constant gene segment for light chains and 5 different constant regions for heavy chains. The rearranged DNA in the mature B cell is transcribed into messenger RNA and translated into the light chain of the antibody. DNA sequences for the variable regions of each heavy chain are assembled by the random selection of a V gene segment, a D gene segment, and a J gene segment. The genes for the constant regions of heavy H chains have Greek letters designating IgG, gamma, IgM, mu, IgA, alpha, IgE, epsilon, and IgD, delta. The DNA in the mature B cell is transcribed and translated into the heavy chain protein. The light chains are then attached to the heavy chains to form the functional antibody. Because of the large number of ways in which the genes can be rearranged, many different antibodies can be produced.
So it's very similar to the lottery, right? Where you have a set number of numbers you can choose from, right, when you play the lottery. So we have a set amount of DNA, and we pick numbers at random of segments of that DNA to make our antibodies and our T cell receptors, so that each B cell and each T cell is unique. Now, so they're showing you basically different numbers for different genes, right? So that is referred to as gene rearrangement. And they literally change their DNA. Like they cut out all these other segments they don't use. Right? It's just what they picked is what now, that's all they can do. Does that make sense to you guys? Right? So that's gene rearrangement. Oh, shoot. I put the wrong title. I'm alright. I was doing it quickly. Imprecise joining they don't show you in the animation. This is what I'm going to try and show you in that. Combinational associations is when you combine things, right? So remember, it's the combination of this heavy chain and the light chain that determines the antigen binding site for this antibody, right? So it's that combination of heavy and light chains that also creates variability, right? So you have the variability first of what it picked for that chain and what it picked for the other chain out of all the choices. And then it's the combination of the two chains that determine that antigen binding site for the antibody. Still with me? Okay, so we still are limited in the number of copies. Say there's about 300 different V's, and as you can see, only 5 different J's. What's the chances that another B cell also picks these same numbers? Could it happen? Yeah. But there's one other step of variation they don't show in this animation. And that's imprecise joining. So that's what I'm trying to show here. Let's see if I need to change this at all, other than the title. So imagine this is a V gene segment. This is another D, and J. Or let's say it's different B and J for like the light chain. Doesn't matter. These are different segments, right? Do you see how I have them blocked out? So this is one segment. Say it's V1. And this is V3. And this is V4. And it decides to put 3 and 4 together. 1 and 4 together. Now look at the actual code though. This is what I want you to focus in on. So do you see the code here in this block and the code in this block? So these two blocks were picked to be put together. But did they take the entire block? No. Do you see that? Right? They took A, G, T, C, A, but it omitted these two T's. For this block, it took the G, G, C, C. It didn't take the two A's. That's imprecise, right? They didn't take the whole block, the whole gene segment, and it joined them together. But imprecisely, they didn't take the whole thing. So that creates variation too, right? That protein is going to be a little bit different than if it had taken the whole block. So even if they pick the same segments, they may not take the same portions of those segments and join them together the same, right? So you could do this same segment, another B cell could pick these two segments, but maybe take the whole segment. Right? So that's one B cell picked both of these segments but joined it together this way, where another B cell picked this segment and joined them together just intact, not eliminating, not doing it imprecisely. So even though they picked the same segments, this guy didn't do it precisely, this guy did. So they're going to be different. Even though they picked the same chunks, same variable regions. Does that make sense to you guys? Yay, I might have succeeded on this one. Alright, so I will make a copy of that one slide and post it on Canvas for you guys. Alright. So during their development, right, they go through this. Both B cells and T cells, they literally rearrange their DNA, right? And so that they get their own unique receptor that they produce. And remember, they're going to be checked out. We don't want anything that recognizes our self as going through and making it through this process. And they do have to be able to communicate with the cells within our body. So that's a much more specific interaction, and we have two different major lineages of T cells, right? We have the helper T cells, which are CD4, right? Remember 4H. And then the cytotoxic T cells, which are CD8 T cells. And I always do another play on words for that. For the cytotoxic ones, they basically go and eat other cells or cause the destruction and death of other cells. So the number 8 sounds like eat, right? I ate something. Eat. So that's how I remember that one. And remember, we watched that process. That happens in the thymus. Where for the B cells, their development happens right in the bone marrow where they're born. Strangely enough, right? Bone marrow starts with B too. But B cells actually are not named after the bone marrow. They're named after the bursa, an organ in a chicken, where they make their antibodies. But it ended up working out okay, because B for bone marrow works too. So I'm not going to play this one, right, in the interest of time. So remember, T cells sometimes need to help the B cells out to go into being activated and proliferating and turning